Yeah. Okay. Yeah, and then let's get started. Yeah, I'm Yuping, and this is a collaboration between me and with Oliver, and our project is called the VaxjoGNN. So it's very informative from the title that we use the Vectro database and use a graph neural network to learn the information in the database. So yeah, let's dive in. So yeah, the motivation is quite simple. Everyone should have to know that predict the vaccine adjuvant to use for specific vaccine development is really, really hard, right? Because the knowledge in the vaccine adjuvant are very scattered. You may find some adjuvant in one paper, but use another name in another paper. So also the data is very, very sparse, heterogeneous, and lacks a common structure. Then and then a very simple question arises. How should we solve this? And we have a very powerful weapon or technology which is ontology. And we have this vaccine ontology database, and which provides very, very structured knowledges. And it contains like 393 vaccine adjuvants. So yeah, it's very have many vaccines, but it's qualitative, not predictive. So how could we make it computational, which means we could input a vac vaccine or adjuvant to it, it could give us some predictions. So this is our research questions. So can we use VO structure to build a predictive engine for vaccine adjuvant selection? And yeah, so now we want to introduce, if we want to utilize the ontology structure, the first thing is what structure it really have, right? So yeah, this is a graph we borrow from Leo, who builds a Vex KG, which is a knowledge graph built upon the VaxJo database. So in there, you could see that it have many, many relations, branches between many kinds of entities, like pathogen and reference and adjuvants. So it's very, very structured. And yeah, and this is a concrete example showing this. So without ontology, we know that CPG is a quite uh, have a many, many kind of variants. And what are those, right? But with ontology, we could do a very detailed search or selection within it. And on the right corner of this slide, you could see that I typed CPG in the keyword search, and it has many, many detailed dropdowns. And the very roughly search told us that there are 13 kind of CPG vaccines, right? And this could be definitely leverage our model or our methodology. And also, the VaxJo is a database, right? So it's have relational schema, which is a very basic paradigm of relations. And this is one example, and we could use this to show that why our graph or our algorithm could work in a very, very high level way. And we have one kind of vaccines, and we could have disease. So this have a relational branch, and this vaccine has a adjuvant. So in the database, there has adjuvant relation. So link it towards to the CPG 1018, right? And to further deep dive in, there's a subclass of CPG 1018, right? TLR9 and TH1. Those are things that we could definitely use. So continue on those labels, right? We could have yield class and TLR pathway and TH1, TH2 biased information. So this mechanism from very curative view metadata, we could use it to like boost our model's performance. And so with all those structures, the next question is how to design some kind of algorithm to learn those structures. And yeah, from the presentation title, right, we use the graph neural networks. And to give a very brief introduction, that the graph neural network is kind of state-of-art AI models for learning from graph structured data. And a basic explanation of how it works is that it has many nodes, and one node, like the previous vectorial graph that represents one kind of entity, like adjuvants or like kind of disease. It learns information or learns knowledge by aggregating, like collecting information from its neighbors. And in the final layer or in the last one pass, the node learns the topological context. In our cases, it's the ontology information within the network. And in this structure, yeah, this is a rough structure of how GNN works, like the messaging passing for node 1 to its immediate neighbor, like the node 2. And then we want to do prediction, but precisely what is it, right? We formulate it as a top K ranking task. The intuition here is that since we have a very large span of the adjuvants, so what we need our model to do is to rank them.
He has frozen.
Yeah. Did we lose him?
I believe so.
Hi, Yiping. We cannot hear you now.
Me?
Oliver, do you want to say something about this presentation?
Okay. Yeah, I think. Yeah, maybe I can. Yeah. So basically, yeah, Yiping, is a college student in the major of computer science. Yeah, from the Hong Kong University in Shenzhen. So he developed the GNN method to predict the vaccine adjuvant selection. So he used the Vegas knowledge graph that our lab has built up. So this one now is about some more specific detail on how it work out. So this slide is about the ranking task. So so it has input. Hi, Yiping, are you available now?
Yeah, it seems I have some connectivity issues back then. Sorry.
Okay, so go ahead.
Yeah. Yeah, we can hear you now. Please continue.
Yeah, very, very sorry. Yeah, the internet is on and off for a while. Yeah, let me back to where I'm then. Yeah, let's start from the problem formulation. So basically, we want to give the prediction or the ranking of a set of adjuvants. So yeah, and the expert then can choose one from these rankings. And the next is how we construct the knowledge. And this is basically the VaxJo database, right? We have the vaccine and have the adjuvant, have the platform, have the disease, and we have those nodes and edge types, which is the relations, and we could learn from these. And yeah, next is how we really learn from the text and the structure. So the first, we need to initialize the node, which is those entities. And we use the SafeBird, which is a biomedical language model, which could encode the node's name and the text description to a feature vector. And this could be read and learn from to the model. And then we will use a simple encoder to project this. And the next is graph propagation, which is how the node learns from its neighbors. And we're using a technique which called approximate personalized page rank. This algorithm is inspired by Google's page rank technique, which is how Google back in the 2010, like how they recommend those web page for the user when user searching some information. So below is the formula. We have the initial state, and it basically is update it iteratively from the past state to the next one, and then it reached the final state. And in the right column, there is a pipeline or a graph showing that we have initial state, and after the graph population, we go to the final state. And we have mentioned that we have some like extra information like the TLR and TH1 and TH2. So in order to give the like the richer information on the signal. So we add those to the model so you can learn much more better information. And we will do a ablation study after this to show that this is really helping. And the last one is we know that the model learns some information about some adjuvants or some vaccines, then how to really rank them. So we have two scores. The first one is interaction score. This is the very basic information, like the adjuvants and what relation it has, and some diseases. And the second, the latter one, is the mechanism prior, which is the TR9 or TH1, TH2, those specific information we provided to them. So we get to the last part, which is the training objective. For the model, like it gets an entire list for a given disease, like the first adjuvant and second one and the third one. And it will predict a rank list, and the model will compute a loss based on this one. So in the L theta, it's the addition of each kind of the prediction, and then it will give a total loss. And to compute this loss, we use a differentiable approximation in order to optimize the model. And we also add a regularization term to prevent overfitting. And in the right column, we have a figure to show this. Suppose that we input brucellosis as the query, and the model will give a ranked list, some kind of adjuvants. Later on, the model will give a list-wide loss, which is the loss in the left corner. And then the model will compare this kind of loss with the ideal ranked list, which directly comes from our database. And then we will get some knowledge, which is called gradients. We will use that to update our model. And yeah, this is a high-level overview pipeline for our ranking list. So we have an input here, and the input will feed into the predictive model. And the model will get the knowledge foundation from our backbone, which is the VaxJo or violin, and they will go through a list of process to give the final answers. And yeah, the next part is the results. First, we do a very simple comparison, like showing why the VaxJo is a very good way to learn those structure. We compare our Adjuvant model with the text base, which is the text mining techniques. And yeah, the key takeaway here is our model shows a very large improvement, 5.4 times over a random selection and 1.5 times higher improvement over a text similarity, which is a very simple variant of text mining for unseen disease. And so what, right? This really shows that the ontological structure or the VaxJo database is really good, and the ontology structure can be used to learn very powerful model and give very useful predictions. And next is ablation studies. So we basically test on some techniques we used in our methodologies. Specifically, the baseline here is that when doing listing, we simply use the information, very basic information from the VaxJo, which is those adjuvants and disease, those very, very simple information. And we train a model based on this. And then we tested it, added the VaxJo classes, which is the TR9 and all those kind of extra information. And the model learns something more. And then lastly, we added a better loss, which is to get the better optimization for the model. And it also gains a little bit. So the key takeaway here is our modeling choice are crucial for the performance. And for the rigorous, we do the bootstrap testing. Oh, and here is a case study. So given the previous some abstract formulas, we want to show that use a concrete example like the brucellosis as example to show that how our model really work under the hood. So the panel A is that the model's top 9 output of the brucellosis. So and the blue bar here is that the top 4 adjuvants really matches with the real world adjuvant's name. So which shows that the model predict kind of well in this kind of cases but one reason here is brucellosis itself is a really well studied disease. So yeah, it's not surprising to get this kind of results. And the next one is the panel B, which shows that how models ranking align with the real ranking in the database. And basically the model shows that it do really well in the top 5 rankings, but the performance kind of worse in the later rankings. And and we have another 2 testings. The first is the panel C, which we want to check that how model is confidence on making predictions. And in the x-axis is how confident the model is. So the 0 is the model itself thinks this kind of adjuvant has 0% for the disease, and the 1.0 shows that it's very highly confident. And in this model, it shows that our model is kind of overconfident. So don't believe that a model tells you it has 100% confident on one kind of adjuvants. And on the panel D, it shows how our model compared to the baseline model, which is only trained on the very basic VaxJo information. And the green bar shows, and it basically works like this. So we both fit the brucellosis to model. One is baseline model and one is our target model. And we compare the top 10 list of rank they provide. So the green bar shows that the target model moves some of the predicted adjuvants to the top, and the brown bar shows that it moves some to the bottom. And at the top is CpG DNA vaccine, which is a kind of famous vaccines. And this shows that our target model is really outperformed the baseline model. And yeah, here is the conclusion part. So yeah, this is kind of our mind flow. The first is the challenge. So the adjuvant selection is really, really hard. And what we do, right? Actually, I think the most important thing we do is we use the VaxJo database and use its ontology structure. So we design a recommendation task and developing an end-to-end GNN. And next is we use some list-wise training objectives and some technical details to solve the optimization problem. And the finding is we do outperform those with no technology ontology structures. Yeah, and there's the limitation and future directions. Limitation is still, although we have the VaxJo database, but the data is still kind of sparse. And there's still some factual errors or the drifts in the VO identifier. So sometimes it will lose some informations. And also the biological structure is still kind of incomplete. So maybe we could fill that up in the future. And this connects to our future directions, like the deeper data integration and more ontology-aware models, which means we could use even better graph neural network to learn those information. And the last, we could use also the better model to learn those things. Yeah, and acknowledgements part, yeah, thanks. Oliver used the funding from NIH grant. And also I want to thank Leo for his useful discussion on VaxKG. Yeah, and thanks. And yeah, I could stop sharing.
Yeah, thanks, Yuping. Any question from the audience? If not, maybe I have a quick question, Yuping. So I know in
Yeah.
general for this type of system, like for prediction and ranking, it
Yep.
is always hard to evaluate, right? Like you can evaluate based on the known thick notes, but for anything new, you don't know if it's true or not, right? So I'm just curious if you have any insight about like how do we know if anything new we couldn't find from prior knowledge? What would be the next step to make sure that the machine learning system tell us the truth?
Yeah. Very good. Yeah, actually, I have to admit this is a really, really hard problem. And in our very specific case, our method to detect whether the model is discover something new is that split the data set, right? So we only train some of the vaccines to fit the model, and we test the model on those settings that it never sees. And yeah, I think you reveal a very good, and I think kind of bad results is that maybe those models could never do what you said. And I think at the end of the day, we still need to collaborate with human experts and to verify whether the result is true. So I think that's the point why it's really valuable to build a network on those VaxJo database, because this is the knowledge that really verified by human experts, right? Yeah.
Thank you.